we're really happy to welcome Dr. Haley Dresang. She is an assistant professor at the University of Wisconsin-Madison. Haley earned her PhD in communication sciences and disorders from the University of Pittsburgh in 2020, working with Mike Dickey and all the other lovely folks over at Pittsburgh. And then she received a T32 grant to work as a postdoc at the Moss Rehabilitation Research Institute and the University of Pennsylvania School of Medicine with Laurel Buxbaum and Roy Hamilton as well. And now she has her own shop at the University of Wisconsin-Madison, where she investigates the neuroscience of language and neurological disorders that affect communication. The mission of Dr. Dresang's lab is to improve the lives of individuals with communication difficulties by investigating neural substrates to cognitive and linguistic factors, as well as how those systems change and adapt with injury. So that's all very relevant to our C-STAR series. She also works on improvement of clinical assessments and treatments for language recovery after stroke. Dr. Dresang has publications on structural connectivity and other predictors of language performance and recovery from aphasia, plus psycholinguistic aspects of lexical, semantic, and event representations. But not only that, Haley is also a ballet instructor, a kickboxer, and a songwriter. So any questions you have about those, hold them until after the talk. And for now, I'm going to give the floor to Haley Dresang.
Thank you for the lovely introduction and for the invitation to be here. I want to just start by thanking you for the opportunity to share my work with all of you, and thank you for attending today. Today I will be sharing two different studies from my postdoctoral work at the University of Pennsylvania in the Laboratory for Cognition and Neural Stimulation. I also then want to take some time to discuss some of our plans for future directions in this line of work here at the University of Wisconsin-Madison in the Weissman Center, where I am now. So I want to start first with an overview. I'll introduce transcranial magnetic stimulation for anyone who is not familiar with it, as well as some of its uses in aphasia. I also will discuss some individual variability that we see in brain plasticity and talk about why this is a topic of interest for aphasia recovery and rehabilitation. I'll talk about two different studies which show some examples of this line of work. These are both from my postdoctoral research period. So study one will talk about ways that we can use TMS to enhance language outcomes in aphasia. And study two focuses on TMS as a means to predict language abilities in individuals with chronic stroke aphasia. And then finally, I'll wrap up by sharing some future directions to integrate and extend this line of work moving forward. So as a brief introduction, transcranial magnetic stimulation, or TMS, is one type of non-invasive brain stimulation. In TMS, we use a sort of figure-8 shaped coil that generates a high current magnetic field. And this is used to target sections of brain tissue more or less the size of your thumbnail. And based on the sequencing of these magnetic pulses, we can either excite or inhibit neural activity in this specific region. And we can use TMS in a couple different ways, and I'll talk about these today. So one way we can use TMS is as single pulses, and this can be helpful to measure brain excitability, like in motor evoked potentials. And I'll talk about this in relation to some of the biomarkers that we can look at for associations with aphasia severity. We can also look at TMS as a run or sequencing of pulses that can be used to temporarily alter the excitability or to measure changes in cortical excitability. And we'll talk about this in relation to studies that use TMS as an adjuvant to speech-language therapy in aphasia. So I think this audience is probably familiar with aphasia, but very briefly, aphasia is a disorder of expressive and/or receptive language, and it often occurs following stroke to the left cerebral cortex. So we know that aphasia severity and the potential for language recovery are highly variable and difficult to predict. So in trying to determine a prognosis in aphasia, this typically requires consideration of many interrelated, patient-specific, and stroke-related variables. So for example, some common prognostic indicators of aphasia severity include things like age, time post-stroke, and total lesion size. However, these standard prognostic measures don't typically include individual differences of neuroplasticity. And we think that these are really critical variables to examine more closely because we know that neuroplasticity, repair, and reorganization is going to be really important for the functional language recovery process. So we also see this immense variability, both in terms of natural spontaneous recovery from a stroke or brain injury, but also we see this immense variability in response to different types of treatments. So both behavioral language therapies and neuromodulatory interventions like TMS. And in fact, we even see in neurotypicals, neurologically healthy brains show substantial variability in the degree of excitability, plasticity, and responsiveness to approaches like TMS. So today I will share two studies that show examples of how we can investigate these types of variability and maybe even leverage some of this variability in different ways to help us better understand the underlying mechanisms at play, and also to eventually better select candidates for these different treatment approaches. So in the first study, I will talk about looking at behavioral predictors of the effectiveness of TMS as an adjuvant to speech-language therapy. And second, we will look at examining some biological predictors, including TMS-derived measures of plasticity that are associated with chronic aphasia severity. So with that, I'm going to start with study one. So this uses TMS as a method to hopefully enhance the effectiveness of aphasia therapies. So here we will consider, in particular, individual differences in terms of different stages of naming deficits to see whether baseline characteristics of aphasia might help to predict the effectiveness of TMS on aphasia treatment outcomes. So this may be helpful to us in selecting individuals or characteristics of individuals that are most likely to benefit from this specific treatment approach. So I won't go into the weeds here because I could give a whole talk just on the body of evidence here, but there is a growing body of evidence showing that TMS can enhance the effectiveness of language therapy and can improve outcomes like naming abilities in individuals with chronic aphasia. I do want to say, though, this is a very large parameter space that we're talking about. There are many factors that we can select and control for, both on the TMS side as well as in terms of the speech language therapy approaches. So for this study, I'll specifically focus on naming deficits because these are common across aphasia classification and severity levels. The study that I'll talk about is also focused on low frequency or inhibitory forms of TMS, specifically to the right pars triangularis, which is part of the inferior frontal gyrus. And the reason that we've chosen this specific TMS approach is based on previous work by both our group and others that has shown that inhibitory TMS to this region tends to enhance naming improvements in many people with aphasia. Furthermore, a few studies have shown that these benefits not only have longevity, but they may actually increase over time, even in the absence of ongoing therapy. And so this is an interesting finding that we need to explore more, but we're sort of thinking that this might be part of the reason this might occur is due to TMS's ability to enhance use-dependent neuroplasticity, which I'll come back to in a few minutes, because this is also a targeted mechanism in certain forms of speech-language therapy as well. So even though there are these promising effects that we see in the literature for TMS supporting speech-language therapy, there's still a lot of variability in the effectiveness of TMS on language recovery, and it remains pretty unclear which individuals are going to be good candidates for different types of TMS treatment approaches. So one source of variability that we see is in an individual's linguistic strengths and weaknesses. A previous study that used this same specific TMS approach indicated, or their findings indicated that this specific TMS approach may work to improve language outcomes, specifically by helping to strengthen the phonological stages of lexical retrieval and language production. So this sort of motivated this study to test how semantic and phonological errors at baseline naming performance may help to inform the degree to which TMS may be effective on the language treatment outcomes. So for this study, we used semantic and phonological deficits derived from the Philadelphia Naming Test, or the PNT, which is an assessment of object picture naming. So there are these great computational models that have been developed to relate an individual's pattern of errors on the PNT to the integrity of semantic and phonological stages of naming. So for example, if you were to see the picture of a cat here, but say dog instead, this would indicate an error between these semantic and lexical stages of lexical retrieval. In contrast, if you said tat instead of cat, that would indicate an error between the lexical and phonological stages of naming. So we refer to the integrity of each of these processing stages as S-weight and P-weight parameters, which then correspond to the pre-treatment semantic and phonological abilities of each individual. So our research questions specifically here were, first, does TMS to right pars triangularis in conjunction with speech-language therapy improve long-term naming outcomes more than speech-language therapy alone? And our second question of interest is, does the baseline semantic and phonological ability of each individual influence the effectiveness that TMS has on their long-term naming? So this is part of a larger clinical trial led by Branch Coslett and Roy Hamilton. So this is an ongoing, double-blind, placebo-controlled investigation. PNT is actually the secondary outcome in the grand scheme of things. But here we have participants who are randomized into one of two arms of a treatment. So either receiving active TMS with speech-language therapy or receiving a sham condition with speech-language therapy. And these individuals are stratified by their aphasia severity and randomized in a 2-to-1 ratio. So all of our participants undergo the same baseline behavioral and neuroimaging testing. They then receive 10 treatment sessions over the course of 2 weeks. At the beginning of each treatment session, TMS is delivered. So for our active group, it is delivered at 1 hertz at 90% of motor threshold for 20 minutes. Then we have the sham group undergoes the same protocol, except that we use a sham coil. So it makes the same sounds as the real coil, but it doesn't actually deliver any stimulation. So our sham group is really showing only the effect of the speech-language therapy. So following our TMS at the beginning of each session, each participant then has an hour of speech-language therapy, which I'll talk about in 1 second. And then after these 10 total visits, we have people come back for 3- and 6-month follow-up visits. And these are the time points at for which we're looking at those outcomes in this study. So for the speech-language therapy component, we are using constraint-induced language therapy, which applies some of those principles I alluded to earlier of use-dependent learning. So these include things like massed practice of conversational language, constraint of alternative forms of communication in favor for really promoting the use of spoken output. So, discouraging, not allowing people to use forms of communication like gesture, drawing, or writing. And the actual paradigm of this treatment is a dual card task game, similar to like a Go Fish card game. Furthermore, we modified the approach in a couple ways. So first, we allow individual therapy with the examiner being our research SLP, who acts as the communication partner. And we have systematic levels or scales of difficulty that a participant can progress through. So this starts from naming the agent noun and verb to production of canonical, modified, and non-canonical passive sentences. So moving from the runner is winning the race to specifying the color of the runner who is winning the race, and then ending with the passive, the race is won by the runner. So the goal of this is that we are engaging multiple levels of the language system, which does include both those semantic and phonological stages required for successful lexical retrieval. So in the current cohort, we have 20 participants who all have chronic left hemisphere stroke. 13 were randomly assigned to the active condition and 7 to sham. Importantly, we do not have any significant differences between these groups in terms of their baseline overall aphasia severity, their overall naming ability, or either of those parameter weights. So for our analysis, we ran generalized logistic mixed effects models, and we ran 2 models, one for each of the follow-up time points, so the 3-month and the 6-month post-treatment times. Our outcome measure was trial level naming accuracy at each of those time points. And we had fixed effects for the TMS condition, so whether people were in the active or sham group, their baseline S and P weights. And we were not we were underpowered with the sample to examine, full three-way interaction. So we examined these two-way interactions between each parameter and the stimulation condition. So we also had random effects for items and subjects here. So looking at our research questions, what we're interested in finding are a main effect of that TMS condition, as well as interactions between TMS condition and each parameter weight. So in answer to our first question, we did see a main effect of TMS condition. So this analysis sort of confirms the advantage of an active stimulation condition for both time points, the 3 and 6 month post-treatment, suggesting that participants in the active stimulation condition had higher naming than those who received sham at both time points. Furthermore, the effectiveness of TMS that we see here also interacted with both the semantic and phonological weights measured at baseline. So these are what these interaction effects look like broken down. This slide shows at the 3-month post-treatment time point. On the x-axis, you can see each parameter weight at baseline, semantics on the left and phonology on the right. And our y-axis shows the modeled outcome of % accuracy on naming. The dark blue line shows the active TMS group, and the gray line shows the sham group. So as expected, we can see by this positive slope in both figures that higher baseline S weight and higher P weight are both associated with greater post-treatment naming accuracy. We also see these robust interactions where you can see an advantage of the active TMS condition was greatest for individuals who had a lower baseline S weight or a sort of medium to high baseline P weight. And so this is what we see for the 3-month post-treatment time point. Looking at 6 months, we see very similar interactions. There is a slightly greater magnitude of each of the effects here. One limitation I want to sort of point to here is because we have a limited sample size, we don't have the greatest sampling across the full range of the parameter space here. So for example, here it looks like people who have lower P weight may not benefit from TMS, but in reality, we have a very limited sample size that falls within this range, so it might not be enough data for the model to really work with. So that's definitely something that we're keeping an eye on for future work. So to summarize the findings from this study, we did find that this form of inhibitory TMS to right pars triangularis prior to constraint-induced language therapy did improve long-term naming ability more than CILT alone, so more than that sham condition. And we saw this at both the 3 and 6 month time points. Second, we found that the locus or the stage of naming deficits at baseline informed the long-term effectiveness of TMS. So although this is quite preliminary evidence, these findings suggest that TMS was more effective for individuals with lower S weight compared to higher S weight, and that TMS may be particularly beneficial for individuals who had higher P weights or stronger phonological abilities at baseline. Now, again, this is likely going to be specific to this particular treatment combination of the TMS and CILT protocols that we used. So the larger study for this is still in progress. So we do hope to report a larger sample with more sophisticated analyses, such as examining some potential trade-offs between the S and P weights, or looking at shifts that may happen across error types throughout the treatment process. Ultimately, we definitely expect that different TMS targets and treatment protocols should be recommended for patients with different patterns of language deficits. But nonetheless, this study is one example of how we can use TMS to potentially facilitate effects of speech language therapy for aphasia. So with that, I'd like to transition to study number 2. And here we employ TMS as a means to index individual neuroplasticity, which can be used to inform models of aphasia severity and perhaps guide future prognostics. So remember that we can use a run of TMS to alter neural activity like we saw in the previous study. But we can also use single pulses of TMS to help us measure cortical excitability. And so that's what we're using here. And in particular, we're using motor evoked potentials, or MEPs, which are more frequently examined in relation to damage and recovery of motor function. However, we here are exploring MEPs as a predictor of aphasia due to evidence that MEPs can index domain general individual differences in cortical excitability and in neuroplasticity. There is some existing evidence already that cortical excitability, when measured in acute stroke, can improve predictions for chronic aphasia severity when controlling for initial severity and lesion size. In our study here, we're looking specifically at using MEPs to index 2 different things. So first, MEPs can be measured after those single TMS pulses over motor cortex, and this provides a measure of baseline cortical excitability. Second, after performing a repetitive TMS protocol, such as the one we saw in the last study, we can measure MEPs again using these single pulses to get individual responsiveness and calculate a difference score from pre to post stimulation, which gives us an index of plasticity of the system. So I'll refer to this as stimulation-induced plasticity. In the study, we also looked at a third biomarker of plasticity, and this one is a genetic factor called brain-derived neurotrophic factor, or BDNF. And BDNF is a neurotrophin, which is a type of protein that is really critical for neural repair and plasticity. So more specifically, BDNF exhibits activity-dependent releases at synapses and modulates long-term potentiation and long-term depression processes, which are sort of the signatures for neuroplasticity. So the BDNF gene has a common polymorphism where individuals will have 1 or 2 met alleles instead of having a typical val-val genotype. This is relatively common. We see these met allele carriers are about 30% of white or non-Hispanic individuals. This does vary by ethnic background, so it's up to 70% in Asian individuals. And there's evidence that carriers of a met allele show deficiency in activity-dependent release of BDNF, meaning decreased neuroplasticity. So as a result, met allele carriers often exhibit reduced hippocampal volume and episodic memory storage. Furthermore, met allele carriers show greater post-stroke impairments in learning, memory, and motor function compared to homozygotes with the typical val-val genotype. So this has been studied in much greater depth in sort of the broad stroke literature, but the effects of BDNF polymorphism on language recovery are not as well established. So we are by no means the first to examine BDNF in aphasia, but there is somewhat sparse and inconsistent evidence regarding the effects of BDNF on aphasia. So one set of findings showed that BDNF polymorphism did not predict aphasia severity or naming improvements following treatment in the acute stage. However, any potential influence of BDNF polymorphisms could be too subtle to detect during this earlier time frame. This study also did not control for factors like age or lesion size, which could account for additional variance in aphasia severity. Another set of findings from Julius Friedrichsen and colleagues demonstrated that MET allele carriers did show greater aphasia severity and naming impairments, even before an intervention was administered. So notably, these studies differ in terms of the phase of recovery their participants are in. So any discrepant findings that we're seeing could reflect potential differences, for example, in spontaneous recovery that could be associated with the BDNF polymorphism. So in our study, we also tested participants with chronic aphasia. And due to this evidence, we predicted that the BDNF genotype would account for additional variance beyond some of those established factors that predict aphasia severity, such as lesion size or age at stroke. Beyond that, our sort of main research question was actually whether these genetic biomarkers and MEP indices of plasticity might interact to improve predictions of aphasia severity. And there is some evidence to support that hypothesis. So there we go. There is evidence that BDNF polymorphism is associated with differences in baseline cortical excitability, which is highlighted here in green, as well as stimulation-induced plasticity, highlighted in yellow. So this is a study done in patients with chronic post-stroke aphasia by Parchour et al. And they found that Val-Val carriers, who are shown in blue, had higher baseline excitability compared to MET allele carriers. They also saw that following stimulation, so here they used an inhibitory form of TMS called continuous theta burst stimulation, where we expect a decrease or suppression of these MEPs. We see this decrease as we would expect in the Val-Val carriers in blue. Whereas our MET allele carriers in red show a lower baseline excitability and perhaps a delayed response, or at least a delayed response in the expected direction that we would hope to see following the stimulation protocol. So we see these major differences based on BDNF genotypes here. Another study by Friedrichsen and colleagues showed that BDNF also interacts with neurostimulation conditions to predict response to a naming treatment. So here they are using transcranial direct current stimulation, which is different, but the general principles are still apply. So what they found here was that Val-Val carriers, again in blue, showed greater improvements following active stimulation than compared to sham, and that this group improved more than the MET allele carriers, regardless of the stimulation condition. So taking sort of this evidence together, we're seeing that BDNF genotype and non-invasive brain stimulation can both influence neuroplastic changes and both can influence language recovery. So we're wondering to what extent these factors may interact to help us predict overall chronic aphasia severity. So in the study, we tested 18 participants, all with chronic aphasia following a left ischemic stroke. 8 of our participants had the val-val genotype and 10 were MET allele carriers. These groups did not have significant differences in terms of age, time since stroke, or lesion volume. We had all participants complete the Western Aphasia Battery, from which we calculated the WAB-AQ, the aphasia quotient, as our measure of aphasia severity. We then recorded 30 MEPs, those motor evoked potentials, both before and after stimulation. So remember that baseline set of MEPs indexes cortical excitability, whereas the MEP change from pre to post stimulation indexes that stimulation-induced plasticity. So for our simulation procedure here, we used a type of inhibitory TMS called continuous beta burst stimulation, which we saw before. And so CTBS uses 600 pulses in 40 seconds here over the primary motor cortex. And so again, the healthy or neurotypical sort of expected response that we would anticipate would be an MEP suppression or a decrease of amplitudes following this stimulation. And then finally, we also collected saliva samples for BDNF genotyping. In this study, we used a backward stepwise linear regression model to predict WAB-AQ, our measure of aphasia severity. And in our model, we included established factors that are documented to be associated with aphasia severity. So age at time of stroke, time post-stroke, and lesion volume. We also included these measures of neuroplasticity, including the BDNF genotype, the baseline cortical excitability, and the stimulation-induced plasticity. And finally, we also were interested in examining those potential interactions between these measures of plasticity. To avoid overfitting our model, we chose to examine interactions specifically between BDNF genotype and these other measures of plasticity, including those neurophysiological measures that we have some sort of suggestive evidence that there may be interactions there. And also looking at an interaction between BDNF and age at stroke. And we were interested in this because age is highly related to neuroplasticity, even in these chronic stages of stroke recovery. So here you can see the optimal model on the bottom of the screen. And on the x-axis of the plot are the model estimates or beta values with brackets plotted for standard error. On the y-axis are listed the fixed effects with interaction effects for each genotype being displayed separately. So once again, we have val-val in blue and MET allele carriers in red. So I just want to draw your attention to the top of the figure here, where we can see that BDNF polymorphism accounts for a large portion of variance in aphasia severity in this sample. This is particularly stark in comparison to the effect sizes for the other demographic or patient-specific variables like lesion volume or time post-stroke. I next want to draw your attention to some of these interaction effects that we found between BDNF and these neurophysiological measures, as well as our age measure. So I'm going to actually break down each of these interactions a bit further. So what we see is interactions between BDNF polymorphism and each of these three predictors of aphasia severity that sort of serve as indicators of neuroplasticity. So first, we'll look at age at time of stroke. So here again, you can see val carriers in blue and metalleel carriers in red. So first, we can see that both groups show lower WAB-AQ as age at stroke increases. However, age did have a stronger effect on aphasia severity in our val carriers. And our post hoc analyses showed that this interaction effect was driven by differences at the lower tercile of age at stroke. So specifically, what we're seeing are individuals with the val genotype are more likely to exhibit better recovery at a younger age, whereas their peers with the met allele carriers below the age of 50 or so are more likely to exhibit worse outcomes compared to the val carriers. The second interaction effect that we found was between BDNF and cortical excitability. So what we see here is that cortical excitability was positively associated with WAB-AQ for val participants, but negatively associated with WAB-AQ for the met allele carriers. And I'm actually going to put a pin in that because I think it makes sense to interpret these two latter findings together. So our third interaction that we saw is between BDNF and that stimulation-induced plasticity. So remember here that we're expecting a negative change or a suppression of the MEP values. So that's reflected in these negative values on the x-axis. So what we saw is that increased MEP suppression, so these negative values here, was associated with higher WAB-AQ for met allele carriers, but did not differ for the val carriers. So following some post hoc analyses and interpreting figures 2 and 3 together here, what we're finding is that val participants tend to show expected MEPs both at baseline and as in response to TMS. In contrast, the met allele carriers show a lot more variability across the board, and the degree to which they show the typical or expected MEP responses is associated with better aphasia outcomes or more mild severity in the chronic stage. So it's possible that these neurophysiological measures provide information about the brain's potential for plasticity, and this is more important for the met allele carriers who maybe don't have some of those neuroprotective advantages that we might see in val carriers. So taken together, these results suggest that indices of neuroplasticity might be incorporated to help us improve models of chronic aphasia severity. And in particular, we found that this common genetic polymorphism, BDNF, interacted with cortical excitability, plasticity, and age. So this study, sort of taking a step back, is a good example of ways that we can use TMS as a tool to help provide information about the plasticity of the brain, which can in turn hopefully provide insight for us into the potential for language recovery in different individuals. So this brings me to some future directions and next steps in this program of research. I do want to start by acknowledging some of the limitations of these studies that might be addressed in some future projects. So we have relatively limited numbers of participants. We also are only looking at cross-sectional samples. So we hope to replicate this work with a larger sample size and to further examine the effects of some of these neuroplastic biomarkers on language changes across time, either from natural recovery or treatment-induced changes. And these questions are of interest and may vary from acute to chronic stages, may also vary in stroke-induced versus progressive aphasias. So that those might be some future directions to explore as well. We eventually hope that this research can inform person-centered recommendations for behavioral and neurostimulation treatments. As I mentioned before, a lot of this parameter space still needs to be explored, looking at different combinations of TMS and language therapy recommendations for different individuals. Two major paths moving forward focus on employing neuromodulation like TMS for aphasia treatment, and also to develop them as biomarkers or prognostic indicators for aphasia. So there are many types of neuromodulation, many types of language therapies in aphasia. We still have a lot of work to identify the types of treatments that are going to best fit different participants and different treatment candidates. We also need future work to help us explore the full power of these TMS-derived indicators of plasticity, as well as other genetic and MRI biomarkers of plasticity that may inform prognostic models or predictions of aphasia. So we have a couple steps moving forward in each of these directions. The first is a pilot study we are starting designed to test how we can maximize plasticity within the semantic network for aphasia treatment. So here we are planning to upregulate activation to important semantic nodes using excitatory TMS now instead of inhibitory, and combining this with semantically related gesture priming. So in this case, both TMS and gestures are non-linguistic techniques that we can use to upregulate the semantic network, hopefully enhancing the activation of residual semantic systems. So following up from study one, this treatment approach may facilitate greater change for individuals who respond to semantic focused treatments. So we expect that individuals who have greater plasticity in their semantic system may likely show improved language abilities. And we can imagine future work can examine parallel methods, for example, centered on strengthening the phonological system as well. Again, trying to tailor the combination of neuromodulation and behavioral treatments for the different types of behavioral strengths and weaknesses that people with aphasia may have. So moving forward on the more prognostic side, we are starting a longitudinal study of multiple genetic biomarkers that are associated with neuroplasticity. Many of these biomarkers have been examined in broad stroke recovery or in traumatic brain injury or other patient populations, but not specifically evaluated in relation to language recovery and aphasia. We're also interested in examining whether these genetic factors may influence changes in structural and functional connectivity that supports changes in aphasia recovery across time. So to summarize, although there still remains and probably will forever be a lot of variability in aphasia recovery and in response to aphasia treatments, techniques like non-invasive brain stimulation can offer some promising steps forward by promoting improvements when used as an adjuvant to speech-language therapy, and by informing the biological basis for models of aphasia, recovery, and severity. So I do want to end by thanking several people. These projects were massive efforts of a large team, predominantly at Penn, but across multiple institutions. So my main collaborators and student assistants on these projects are shown here. I also want to thank our funding sources at the NIH and all of our participants and their support systems. A lot of this data was collected through the height of the pandemic. So I'm especially grateful for all of them. And finally, thank you for the invitation to share this work with you, and thank you for your attention. I'm really honored to be the final speaker of the series this year. So thank you.
Great. Thank you so much, Haley. I'll turn the clock. You don't see me, as long as you can see the room. Thank you so much. That was a really nice talk and a perfect end to our season for C-STAR. But before we end, we have questions. So I want to start by seeing if there's questions from our own room. Seeing none. In that case, I am going to turn the camera. Okay. Here we go. Here we are. So then we have questions online. So the first question is from Hale. Do you think low frequency stimulation of the right pars triangularis and concurrent high frequency stimulation of the left pars triangularis can significantly improve naming? Can you use simultaneous stimulation with your approach?
Very interesting question. So it would be possible to use simultaneous stimulation. You know, one challenge that we have in this population, and we're facing it with this study right now, is using stimulation in the left hemisphere can be really challenging based on where the lesion is in each individual, and not even direct lesion, but how all of the networks may be impeded by the stroke. So I don't know of any work that has done this specifically with low and high frequency stimulation, like bilaterally like that. So I don't have a great answer about the actual feasibility. In theory, it would be possible to do that, just technically challenging.
Great, thank you very much. I have a question from Kirana Tsapkini. Great talk and very interesting work, Haley. 3 questions. 1, is excitation or inhibition reliable with TMS? Can you tell if it is one or the other from the MEP? Do you assume that motor plasticity is equivalent to language plasticity? So that was that was the first question.
Okay. Shall
I read the second and the third first?
Let me try to tackle that first. So is excitation or inhibition reliable? The short answer is not really. There is a lot of variability, and I think that's one of the things that we're seeing in particular with those MET allele carriers, is that we are seeing more variability in not just the sort of effect size and the amplitude of MEPs, but also in the directionality. So for some individuals, we might see increases where we expect decreases, or vice versa. So across the board, as you know, when we look at group level data, they do tend to be reliable, but there's a lot of individual variability in that.
So
I don't know if I answered all of that.
Yeah, no, yeah. So another part of that question was, and that's an interesting one as well, is whether you assume that motor plasticity that you measure with the MEP is equivalent to language plasticity, right? So is plasticity a homogeneous?
Yes, great question. And I think Roy Hamilton would love to talk more about that, because that was part of his rationale for the grant that he wrote for this project. So yes, so the motor evoked potentials are measuring motor plasticity, right? We're just measuring in the motor cortex. I don't think this is equivalent to language plasticity. However, there's a lot of data showing high associations between the motor and language systems, including the neural networks that support them. So really what we're using MEPs here for is almost to serve as a domain general index of plasticity, rather than something that's language specific. So really what we're kind of showing is these measures that have been applied to motor outcomes and motor recovery may be extended to help us predict language performance as well. But I definitely agree that motor plasticity and language plasticity are not exactly the same.
Thank you. Another question from Kirana. Do you assume that areas of stimulation would be specific for each type of naming error? So for example, why would phonological errors diminish if the area is not related to phonology?
Yes, great question. So we would not assume that, I'm just recalling the question, that areas of stimulation would be specific to different types of naming errors, right? So different types of naming errors are likely to be localized to different regions of the brain. So using more targeted simulation treatments based on those loci of behavioral deficits might be a more targeted way of looking at this, compared to, in the study one that I presented was not actually designed to be looking at the semantic and phonological predictors. This was sort of a secondary question that we were looking at. So it wasn't specifically designed for that. I think we were to design a study around sort of different areas of stimulation for different types of naming errors. We would be looking at different targets.
So that is related to a question that I had as well, and I'm sure this is a question that you have discussed or that you've had before or that you've talked about. I'd just love to hear what you think of this. So related to study one, what do you think is the functional mechanism that is suppressed or indirectly enhanced through your right hemisphere IFG RTMS?
Yes, this is a great question. And actually, I have thought so much about it that I have an additional slide. So, yes. So I think we've thought a lot about different possible mechanisms that might be happening here. And one plausible explanation that I tend to like for this is so an explanation for the relatively strong phonology findings that we're finding for this right pars triangularis inhibition. So the right pars triangularis is often presumed to play this more maladaptive role in aphasia recovery. So behavioral improvements following this inhibition are kind of thought to be associated with more posterior shift in the recruitment of regions within Broca's area homologue, so the pars opercularis. So in the left hemisphere, the pars opercularis and triangularis subserve functionally dissociable roles, so phonology and semantics, respectively. So we're sort of speculating that this could suggest that inhibiting the right pars triangularis may confer some of these naming and specifically phonological improvements by prompting sort of behaviorally beneficial recruitment of the right pars opercularis, which then enhances that phonological access process.
Which is not what your treatment is targeting at all, right?
Right, exactly. Yeah, so this is not phonologically driven treatment. So it would be interesting to see how that might even more so give us robust outcomes.
Thank you so much. We do have a final comment / question from Kirana. She says, I'm afraid that for such unbalanced baselines, it's hard to compare the TMS effect. Have you tried combining the two conditions for baseline matched samples?
So the unbalanced baselines in terms of the parameter weights, is that?
I don't know.
Okay, I can follow up with Kirana about that as well, because I'm interested in talking about that further.
Okay, so you can take that to her.
Clarify, yeah.
Okay, thank you.
I also can send around my email to anyone that is interested in discussing further.
Great, thank you so much. I did, if we do have time, then I had a really minor point on study 1. Looking at the way you presented the graphs, I wasn't quite sure whether the interactions that you showed between TMS and those S&P weights, do they apply to the naming accuracy at a specific time point or to the change scores? I would think the change scores are what is most relevant, but it looked as if you had naming accuracy as your outcome measure.
Great question. I think that was a mislabel on my part on the graph.
Right. So it's the difference between
it's the difference between baseline and each post-treatment follow-up.
Thank you so much. Thank you, Haley. That was great. This concludes our season 7 of C-STAR, and we're looking forward to seeing everyone again in August 2023 when we'll be broadcasting on Thursdays at noon. Thanks, Haley, and goodbye, everyone.
Thank you, everyone.